What's up, Chicago? We can do better than that. What is up, Chicago? Welcome, everybody, to TechChicago Week and TechChicago Day. Make some noise for yourselves. All of TechChicago Week is a fantastic collaboration between P33, World Business Chicago, Intersect Illinois, and 1871. And this is really meant to be where tech and culture and opportunity all come together. And it highlights that today at TechChicago Day. My name is Rajiv Nathan, aka Raj Nation, the startup hype man. If you haven't heard of me before, I haven't been loud enough, apparently. I've been helping Chicago's founders for the last 7 years, founders in Chicago, nationwide, and across the world, helping championing their voice through pitch development and connecting them with opportunities to grow. And I came to know about P33 3 years ago when they launched the TechRise pitch competition. And I saw that, and I was immediately a fan. And then I was like, I got to get to know these people. And so I started to get to know them. I got to meet Desiree Vargas Wrigley. I got to meet Claude Simius, Alonzo, Lucy Brennan, and a lot more at the TechRise team. And if I can just for a moment, raise your hand if you know Lucy Brennan. All right, some of you know her. I don't think she gets enough flowers. She does so much for this community. And she likes staying behind the scenes. She's that real G who moves in silence like lasagna. But she does so much for this community to uplift and keep it moving, and we're all so grateful for the work that she does and that all of P33 does. So after being a fan, I became a friend of P33, and then through being a friend, we got to know each other, we've collaborated on different events, and now I enjoy not only being a friend, but being able to stand on the front lines alongside them as we work to make Chicago a Tier 1 tech hub. And not just any kind of Tier 1 tech hub, but one where capital, opportunity, and inclusion do not have to be mutually exclusive. So it all comes together today at TechChicago Day. This is the flagship event of the week. Have you been to the Capital Summit? Raise your hand or make some noise if you've been to the Capital Summit. How many of you played poker? Yeah? Who went to the networking mixer last night? Make noise for that. Still recovering from that mixer, I see. All right. So we're here at the culmination of the flagship event, TechChicago Day. Thousands of technologists and Chicago enthusiasts are coming through today. We've got a stacked lineup on this stage. We're going to be talking tech. We're going to be talking culture. Later on tonight, Joakim Noah is going to be here. Common is going to be here, the man who is for the people. You're going to see a whole lot of great stuff going on this stage and throughout some of our talks as well. There's going to be a breakout stage afterwards where you can ask the speakers specific follow-up questions 1-to-1. And don't miss everything happening on the 7th floor as well. This is a multi-floor event inside this beautiful building. On the 7th floor, we've got the AnitaB.org stage. That's a focus on women and non-binary technologists, the employment opportunities that come along with that, and there's also a schedule of informal meetups happening in our lounges as well. Being on inside this beautiful building today, courtesy of Sterling Bay, I want to just do a quick land acknowledgement. We are on the traditional lands of the Ojibwe, Odawa, Potawatomi, Anishinaabe, Ho-Chunk, Kaskaskia, Kickapoo, and there's more. The Mascoutin, the Menominee, the Muskawa, the Peoria, and the Second Fox. All, we are honoring the lands that they originally inhabited here today. So a round of applause for that. So we pay respect to our elders, both past and present, with that. Now, we can't have TechChicago Day without a pitch competition, right? Can we have TechChicago Day without a pitch competition? No. That's what I like to hear. Okay, so to kick off the action today, we have the TechRise pitch competition where 5 companies across life sciences and biotech are vying for $25,000. To introduce these startups and the person who will moderate, I would like to welcome at this time, Jessica Brown from Sterling Bay. Please welcome Jessica to the stage. Jessica is the principal at Sterling Bay, responsible for overseeing all the leasing efforts across its growing national portfolio in Chicago, Miami, Atlanta, Durham, DC. And she leads the leasing strategy of the Fulton Market and Lincoln Yards. I'm sure you've seen some amazing buildings going up in both of those neighborhoods very recently. Since 2015, Jessica has been an integral leader of Sterling Bay's leasing team, specializing in overall asset strategy, lease negotiation, and place making. So she is going to introduce the TechRise competition today, which is brought to you by Sterling Bay, Bolden Networks, Gensler, Kinema Fitness, Office Revolution, and Powers & Sons Construction. One more time, give it up for Jessica Brown.
Thank you for that intro. Hi, everyone. I'm Jess Brown, principal at Sterling Bay, the landlord and developer behind this building, 1229 West Concord, and the future neighborhood in which it sits, Lincoln Yards. On behalf of both Sterling Bay and our partner, Harrison Street, we welcome you to the building today. We are extremely proud of this building, and we are extremely proud to be hosting TechChicago Week. Just in the past 2 days that I've experienced TechChicago Week and what I will experience today, I think that the event has really embodied the potential for Chicago's tech community. There's 3 themes that come to mind when I wandered around the spaces yesterday and attended the events. It's the innovation, collaboration, and inspiration that can happen in this community when it's brought together. So those 3 themes really come full circle for us, because when we were designing this building just a few years ago, those 3 themes were exactly what was at the forefront of our mind when we were designing the amenity spaces on the 1st and 2nd floor, the vacant floors for future tenants that we're sitting in here today, the interior design, the outdoor space, the views along the river. So those 3 themes have really come full circle, and seeing this event take place today makes us very proud. So I encourage all of you to wander around the building, explore the beautiful spaces that you'll be in today. And if you have any questions about 1229, the building, or Lincoln Yards on a broader sense, please reach out to myself or anyone on our team, and we'd be happy to share and connect further. I'd be remiss not to thank and congratulate the P33 team. You guys are a small but very mighty team, and what you've pulled off here today and this whole week has been wildly impressive. Also want to thank you on behalf of all of Chicago, because what you guys do for the city and the tech community here is really important for the future of the tech community. And lastly, I want to thank and introduce Desiree. I've gotten the pleasure to know Desiree over the past year or so, and she's been a very valuable resource for me, and what she does for the Chicago community is so impressive, so thank you. Desiree Varchas Wrigley is the Chief Innovation Officer at P33 and an Executive Director at TechRise, and soon to be founding GP of Velocity Catalyst, a new $50 million Chicago land-focused fund. With that, here's Desiree.
Thank you so much. And Raj stole my line about thanking Lucy and the team for everything that they've done this week. It's really impressive. Truly, we are a 3 or 4 person founders team inviting thousands of people from across the country to this. So thank you so much for all your hard work. So for those of you who are unfamiliar with TechRise, we are an initiative started in partnership with Verizon about 3 years ago to help support overlooked founders across Chicagoland. And each week, founders take the stage, mostly virtually, so we don't do this live all the time, for a chance to win $25,000 to $50,000 to go build their dreams. And over the last 3 years, we've welcomed 352 startups to the TechRise stage, and our founders have gone on to raise almost $160 million in follow-on funding, which is up from just $9 million in 2019. So it really has been hugely impactful. And we are excited to continue to do that with the support of the Minority Business Development Agency, who hopefully is here supporting us with the Capital Readiness Grant. And we also have several other sponsors that I'd love to thank for today. So today's prize money is brought to us in part by Sterling Bay, Bolden Networks, Gensler, Kinema Fitness, Office Revolution, and Power Construction. So thank you all for making this possible too. All right, and so today what we're going to do is we're going to have founders take the stage. They'll have 4 minutes to pitch, followed by 4 minutes of Q&A with the judges, and then the judges have the very difficult task of going and deliberating to figure out a winner in about 10 minutes. And while they're deliberating, Michelle Hoffmann and Tom Denison will take the stage and talk a little bit about what's happening in biotech and life sciences in Chicago. All right, so with that, I'm going to welcome our judges to the stage. First, we have Margarita Chavez, venture partner at Wellington Life Science Partners, a seasoned biopharmaceutical executive with over 25 years of deal-making experience in the healthcare industry. She's currently a venture partner at Wellington and serves on the board of Neuron Pharmaceuticals. She's also an investor and advisor to a number of early-stage biotech companies in the Chicago area, and a founding member and, until recently, managing director at AbbVie Ventures, where she was responsible for over 24 investments ranging from seed, spinouts, to crossover rounds in companies of various stages, including Elector Pharmaceuticals, Charisma, Morphic, and which was recently acquired by Lilly for $3.2 billion. She's also on the board of P33. You can come join us. And next, I'll introduce Julia Monfrini-Pieve, founder and managing partner at Pace Healthcare Capital. Pace Healthcare is a venture capital firm investing in digital health and healthcare technologies based in Chicago. As a seasoned institutional VC and private equity investor, she's deployed $1 billion of capital in successful and transformative healthcare businesses with a demonstrated track record. And then, woohoo! And next, we have Nancy Sullivan, CEO and Managing Director of Illinois Ventures. Nancy leads the Illinois Ventures team in fundraising, investment strategy, and limited partner relationships. A key advisor to startups, Ms. Sullivan assists companies in obtaining venture capital funding, recruiting management teams, and advising on business development and commercialization matters. And prior to joining Illinois Ventures, she served as Senior Director of Business Development for an early-stage biotech company where she led BD and managed corporate office operations. So, welcome. All right, do you have Robert? Oh, and Robert, where? Oh, sorry. And then next we have Robert joining us. I'm sorry, I didn't see you lined up, so I was worried that you weren't here, but you are here. Robert is Director of New Ventures at Discovery Partner Institute and leads business building initiatives at DPI as director of the New Business Ventures team. His experience in technology commercialization includes 15 years as a consultant to 7 universities and 3 Fortune 500 companies. A practitioner of the art and science of entrepreneurship, Bob has been a co-founder of 4 companies and an angel investor in 12 other ventures. He's been a startup interim CEO, interim CFO, board member, board chair, or advisory board member for many of these startups. And collectively, they have raised over $200 million, and 3 have been acquired by public companies. So, welcome. Thank you. All right, and with that, we are ready to get started. So our first founder taking the stage is Rebecca Keate with CellMend Technologies.
All right, thanks everybody for coming out today, and I'm really excited to give my pitch. I'm Rebecca Keate, and I'm CEO and co-founder of CellMend Technologies. Today, I'm going to be talking about surgical wound infections, which are a major problem in the US as well as globally. The median cost of treating a patient who has a surgical wound infection is about $40,000 per patient, and this amounts to over $5 billion in supplemental US spending. Now, surgeons will tell you that only up to about 5% of their patients will experience a surgical wound infection. But if we look closer at this number, we see that surgical wound infections are very common, especially for patients who have comorbidities or health complications that lead to an atypical skin microenvironment. Existing products do not protect these patients from infection. And even for patients that are protected from infection with these products are highly irritating, damaging, and can even cause severe allergic reactions. We need better solutions. Our solution to this problem is a new molecule called panthenol citrate. Panthenol citrate is synthetic, but based on naturally occurring skin-healthy molecules. It's antimicrobial, and in addition, we have synthetic IP protection, So we're protected through a compositional patent. It's also anti-inflammatory, so gentler for skin. And it's also luminescent, which is really interesting because we can use this for enhanced infection monitoring as well as detection. Because of the multifunctionality of panthenol citrate, we performed in-depth customer discovery in order to determine the best go-to-market strategy in our pipeline. From this, we learned that starting with a wound decolonizing or skin conditioning wash, and then moving into the development of an infection detection and monitoring spray, leveraging this luminescence gives us the best opportunity to get traction in the market. With the first product alone, the market is huge. And despite the number of products and the number of times that they're frequently used, no existing technologies actually have the multifunctionality to be compatible with skin pre- and post-surgically. This is primarily due to the significant cytotoxicity of the harsh chemicals that are in these current solutions. In addition, we identified a gap in the market with pediatric patients who are at risk for surgical infections, but don't actually have products tailored to address their needs. For this first product commercialization, we're currently working on developing a formulation, and from there, we'll be able to perform non-clinical testing for our 510(k) submission, which will lead to a clinical study for marketing purposes. Should we receive funding today, that funding will go towards developing that first prototype. We plan to launch our first product halfway through 2027, with the second product following in 2030. From there, our exit window will open, and acquisitions in this space are very large and very popular. Our team is uniquely suited to address this problem, which is more urgent now than ever. Because of the increasing problems with antimicrobial resistance, we need new and more effective solutions to protect the patients who need it the most. I have industrial experience in consumer as well as medtech product development and know how to systematically take a product to commercialization. My co-founder, Dr. Guillermo Amir, is one of the world's leading experts in citrate-based biomaterials. They're actually developed in his lab, and he has a record of successfully commercializing this technology, so we're well equipped for this challenge. Overall, we aim to improve the quality of life for all patients, even the most vulnerable, and panthenol citrate is uniquely suited to accomplish this goal. Thank you for your time today, and I'll now take any questions.
All right. Bob, we're going to start with
So, as somebody who's had an adverse reaction to one of the products you had in your graphics, I'm very interested in this. One of the big challenges for products like this is distribution and getting into various medical facilities. Do you have thoughts on what your go-to-market strategy will be once you prove your formulations?
Yeah, thank you for that question. So we do plan to try to find a distributor to partner with. And then in addition to that, we're trying to increase our traction and actually be incorporated into CPT bundles, because that's a lot of the times how hospitals actually work with these infection protection strategies. So they package multiple products together. And so our goal is to find different corporate partners that are going to help champion our technology and actually get it into these bundles with the partnership of a distributor.
Nancy.
Rebecca, thank you. Really well presented. My question is the specific of your first go-to-market, which is in that pediatric space. You said the 25K if won today would allow you to get to first prototype. Can you elaborate for that, of what you'll be able to do with the 25K?
Yeah. So we plan on developing the first prototype in partnership with a CRO that is an expert in formulation development. And one of the first steps to do that is we have to do a really systematic analysis of the molecule's degradation and stability. So for example, if exposure to a lot of light is something that is going to cause the molecule to lose its functionality, that's something we need to find out as soon as possible. And the 25K is enough to do that thorough degradation analysis with a CRO.
Thank you. I was about to ask user funds for the $25,000, so thank you for addressing it. Then I'll turn to my next question. How much capital would it take to get to a launch?
For launch of the first product, we estimate it'll be about $4 million.
Thank you. What is your VC go-to-market strategy or your VC fundraising strategy?
Yeah, that's a great question. So in terms of VC, I think what we need to do is develop a lot more data, preliminary data for the next products, like the infection detection and monitoring, so that we can improve the confidence that we're going to be able to develop more products and get an even bigger market beyond the first actual product. Because part of what's really exciting about this technology is that we want to build a platform based off of it. We're not just thinking about a single product or a single problem.
Great, thank you. Great presentation. With the wound care business, it seems like the hospitals already probably have ones that they rely on. And you'd probably have to dislodge those. And I understand there's differentiation. Do you think, and maybe how would you pitch your differentiation to a hospital? Is it material enough that they would dislodge whatever they already have in their system? And then second, if you are able to do that, your IP, what type of IP do you have? I know you have some synthetic type of IP. Is it composition matter patent? Is it just a formulation patent? Do you think it's a type that you could defend if you are able to penetrate that market?
Yes. Okay, so for the first question, in terms of how we'll differentiate to the hospital, it comes down to efficacy. Because these infections are so expensive per patient, about $40,000, right? Even a slight improvement of efficacy or reducing the complications is enough to convince the hospitals, especially if we have doctor support, which is one of the benefits of doing the customer discovery process sooner, is that we can make sure we're developing a product that doctors are going to trust and has the qualities they want to see. So, and then, sorry, could you repeat? Oh, the IP. The IP. So we currently have a composition patent, and we're planning on, as we develop the formulation, enhancing
I have to keep it fair and cut you off, but great job, Rebecca. Thank you so much. All right. And next, we're going to welcome Khalid Alam with Stemloop.
The future is biomanufactured, and it's happening here and now in Illinois. Biomanufacturing is the process of sustainably producing physical goods from cheap and abundant inputs. It's the process that Nature's Find out in the Chicago stockyards uses to produce meat and dairy alternatives. It's the process that LanzaTech in Skokie uses to capture carbon pollution from the air and produce sustainable fuels and textiles, like the jacket that I'm wearing today, which was made with captured carbon. So there's a biomanufacturing gold rush that's underway. McKinsey estimates that up to 60% of all physical inputs into the global economy can be biomanufactured, with an economic impact of $4 trillion annually. Others say this estimate is too conservative. Either way, over the last 2 years, over $29 billion of investments have been made in US biomanufacturing, recognizing it as a critical technology for our national economy and our national security. Illinois is also investing in our future, with over $700 million behind the recent launch of the iFab Tech Hub. Now, despite the major potential and massive investments in this space, biomanufacturing has a few problems to solve. For one, biomanufacturing R&D is bottlenecked by chemical testing. Testing is core to the design, build, test, learn cycle that R&D teams use to bring new products to market. It's how you know if the process that you're engineering is actually working. But traditional methods for testing are far too complicated, too costly, and too slow. They simply can't scale to meet the industry demand. LanzaTech calls testing a definite choke point, and other companies in this space will tell you that testing is a major challenge and that rapid analytics are a critical and urgent need. Stemloop solves this problem. We accelerate biomanufacturing R&D with easy-to-use, inexpensive, and rapid chemical tests that scale. So rather than complicated, costly, and slow, our technology is easy, cheap, and fast. It's a drop-in solution for biomanufacturing workflows with pennies per test cost economics. Our technology enables R&D teams to collect hundreds of data points in the same amount of time that traditionally takes them to collect a single data point. That uniquely positions our technology as having both the high performance of traditional analytical chemistry as well as the scalability of other test-based kits. We can do this because biology is the best analytical chemist, and our team has sort of cracked nature's code on sensing small molecules. So at Stemloop, we identify these mechanisms that biology has evolved over 4 billion years to detect everything from nutrients to contaminants, and we engineer inexpensive, easy-to-use biochemical sensor reactions. We've been generating revenue, building sensors for our partners to address our partners' large-scale unmet chemical testing needs. We discover biosensors using computational biology and bioinformatics. We utilize next-generation nucleic acid design and protein engineering tools. And finally, we can deploy these biosensors as easy-to-use and low-cost chemical tests. We had strong early traction with both government partners like Chain Reaction Innovations at Argonne National Laboratory, as well as commercial partners like LanzaTech and Nanophase Technologies out in Romeoville. But we need your help. I need your support, Illinois' support, Chicago's support, because we're getting ready to raise a $3.5 million seed round. And a win today would catalyze our efforts by allowing us to develop product packaging, to continue to do our customer discovery, and to secure letters of intent. And here's why we'll succeed. We have the world's best team. I founded Stemloop after spending a few years at Northwestern Center for Synthetic Biology.
I'm going to have you cover the rest of it in Q&A. Keep it fair. All right, Nancy, will you kick us off?
Thank you.
Nancy, will you go first?
Thank you so much. Very nice presentation. I had a quick question for you. 4 minutes doesn't give you all the time you want, but you mentioned some commercial partners, plus you also mentioned trying to get letters of intent. So can you give us a little more detail on that commercial partnership and what those actually mean and represent?
Yeah, absolutely. So the commercial partnerships that we've had to date have been service revenue. So using our platform to discover and develop biosensors for their given applications. What we're trying to do now is sort of transition to a more recurring product revenue model. And so we're developing sensors that not just one customer would use, but the entire biomanufacturing ecosystem. So being able to measure things like central metabolism or branch point intermediates in metabolism, for example.
Thank you.
You're welcome.
Great presentation. Congrats on all the success. Could you tell us more about who your typical customer is and your go-to-market strategy?
Yeah, absolutely. So our typical customers are biomanufacturing companies that are involved in R&D. So the customer profile here are metabolic engineers or strain engineers at these companies, where they're trying to develop new strains to produce new products, and as a result, they have to do a lot of testing. As far as go-to-market strategy, we've done a really good job of making ourselves known and prominent within this ecosystem. I think part of what's going to be required here for us to go to market successfully is making sure that we continue to show up at those meetings and continue to sort of share with them the benefits and the features of our technology. Thanks.
So, first of all, congratulations on what you've accomplished. It's quite a lot. But my question is about, like, it seems very capital intensive. How much do you think it'll cost to get to the next inflection point? You say a $3.5 million seed, but where will that get you? And then how much more will you need to get to something that might be transactable or exitable?
That's a great question. So definitely what we do is capital intensive. We've got a great team. We run a life science laboratory. Consumable costs alone are quite high. We're looking to raise a $3.5 million seed round. I don't think we need that full amount to get to market and to start selling products. I think it's a fraction of that. Yeah.
So the world of bioengineered materials is hugely diverse. Is there a subsegment that your technology has gotten more traction in than others? And will that be your first target market, or do you feel like you have more of a broad platform?
Yeah, so I definitely think that we have a broad platform, right? So if you think about this, biology has sensors for everything from ions, right, tens of Daltons, all the way up to 1,000 Daltons, where we're talking about complex polyketide molecules, for example. And we're just sort of scratching the surface of what's possible here. As far as customers, the customers that we have worked with are typically looking to be able to measure their end product. Now, and that sort of requires a bespoke, tailored solution for that particular customer. So what we're hoping to do is sort of shift over to needs that are shared across the industry. So things like central metabolism or branch point intermediates would have relevance across industrial biomanufacturing specifically. Thank you.
So these biosensors, just really quickly, I'm sure you have some proprietary technology in it, and you have IP. How hard would it be for your competitors to come up with something similar that can do the same thing?
Yeah, so I think it would be really difficult, in part because my team, my co-founders, Mike and Julius, we were the first to sort of demonstrate a lot of these technologies. We have a pretty wide IP moat at this time. We've licensed 4 technologies collectively from Northwestern, MIT, and Harvard, and we have filed patent protection for technologies developed internally as well. Yeah, thank you.
Great job, Khalid.
Thanks all.
All right, and next we have Ashima Shukla with Varchas, Biotechnologies.
Okay. So cancer might be just a word for some of us, but it's actually a battle a patient has to go through when they are diagnosed with it. In late 2022, someone who's very close to me was diagnosed with it. After going through an emotional turmoil, me and my husband started to discuss what are the therapeutic options for cancer patients right now. We quickly realized that one of the biggest breakthrough happened in the field was these engineered T cells, aka CAR T cells, which has actually cured several cancer patients. However, they're still not the best options for several patients because there are challenges. These therapy can fail in patients if the cancer mutates itself, if it's a solid tumor, if the T cells become dysfunctional, and there's high toxicity. And on top of it, there's a big manufacturing hurdle, which makes it really difficult to make these therapy accessible to several patients. On that day, I decided that we will overcome these challenges by developing a universal CAR. We have developed a MAS-CAR, which is a modified antibody-specific switchable CAR, where the T cells actually recognizes a special chemical which can be tagged onto a monoclonal antibody and can bring in different types of immune cells at the cancer site to eradicate cancer. We know that when we use this technology, we see enhanced efficacy and about 2-fold more decrease in tumor burden when we compare it to the conventional CD19 CARs in vivo. Here's our timeline, and if we win the TechRise competition today, we will use these funds to do the toxicity studies and bioavailability studies, which will unlock the seed round for us. This field is highly competitive, where there have been made several CAR T-cell advancement. However, how we are unique is because we are trying to bring in different types of immune cells at the cancer site, and which actually eventually increase the efficacy. We are universal, off the shelf, and we have the better T-cell functionality. Even right now, with so many therapeutic options, there are so many patients who are in the refractory phase, and they are left with no therapeutic options. If we are successful, we will be able to solve or help several patients with heme malignancies and moving to the autoimmunity and then to the solid tumor. This is a highly rapidly growing market with a CAGR of about 30%, and this is one area where big pharma actually relies on the biotech for the discovery. And we have seen successful exits early on. And we have the right team to do it. Both Vipul and I are passionate immunologists, where we think that if we bring in the different immune cells at the cancer site, we will be able to eradicate cancer in several patients. And we have the team of clinicians and industrial experts who see CAR T-cell patients, who have developed these technologies before, and we are dedicated to take this technology into clinic to help several patients. With that, I would like to summarize that we have developed a universal off-the-shelf CAR, where we are trying to mitigate the major problem with the cell therapy right now. And our goal is to help different cancer patients. With that, I would be happy to take any questions, and thank you so much for your attention.
Great job. Julia, will you start us? Julia, will you start?
Okay, first of all, you are addressing a real problem. Okay, that I know. So many questions, though. So with this mass CAR, are you going to be able to then do in vivo CAR delivery,
Yes.
meaning in situ into patients? And second, have you tried targeting with it?
So we haven't tried the in vivo CAR. So the thing is that this universal off-the-shelf CAR can be given to anyone, and we are agnostic about the way we want to deliver it. In vivo could also be a possibility that we can give. We have tried right now just some of the targets just to do the comparison with the conventional CARs. We haven't tried delivering in vivo yet. Yeah.
Thank you. And could we double click on product development and efficacy? What type of data do you have right now that shows actually that you are able to address the issues currently with CAR T cells?
Yeah, it's not going back. It can't. Okay. Sorry, I can go back to that slide, but yes. So we did testing in the solid tumor model. We engineered the cell line to express the CD19 and did the head-to-head comparison with the CD19 conventional CAR. And we saw that there was enhanced survival as well as twofold more reduction in the tumor burden when we compared it with the conventional CAR. And to our surprise, that even when we stopped the antibody treatment, which is the modified antibody, several mice showed the anti-tumor response, and we were able to, the tumor did not grow to its size for, after several weeks of when we stopped the treatment. Yeah.
And I'll just continue maybe along the line and go down one more level. You mentioned the 25K where it's going to go towards toxicity and bioavailability studies. Can you give us a little detail on how that 25K really covers that? And then your use of the 2 million tranche 1 in your seed funding.
Yes. So we are right now in the process of starting new in vivo experiments through the CBC Directors Fund. If we win this award, we will combine it with that, and we will collect the tissues to do the toxicity in the heme malignancy. And then we would do the bioavailability of the antibody. We will just collect the serum at the time of when we are doing the in vivo experiment. So we'll just collect the specimens at that time, and we'll use the 25K to do the bioavailability as well as this toxicity. And for the tranche 1, for the seed round, the moment we get that, we will start establishing the prototype, and once we have streamlined the process of the manufacturing outside of the lab, we will go and we will initiate the process development, and we will start packaging the IND studies.
And the CBC grant was $60,000?
Yes.
Thank you.
Yes.
Appreciate it.
Mic drop.
That's okay. So you've identified a huge number of challenges in the CAR T space. But somebody who is maybe a little bit more cynical, that's me, might say that you need to specialize in either one type of cancer or one type of tumor, whether it's specific types of solid tumors first. Do you have kind of a hierarchy of the types of tumors you are going after?
Mm-hmm. Yes. So we want to start with the hematological malignancy because the process is more streamlined, the regulatory path is more clear in that, and with that, we will have the toxicity profile, and we can see if we can move to autoimmune diseases. Once we have the better understanding how the CAR is working in vivo in patients, then we will slowly advance the technology to the solid tumor. We want to start with the ovarian cancer. We have several targets in mind that we can go with.
Perfect. Thank you so much, Ashima. Great job. And next we have Jeremiah Kim with Signl.
"75% of the world got a COVID vaccine the past couple of years. 80% of those people had negative side effects associated with those vaccines. Additionally, 50% of people in the US get their flu vaccine every year. But even with this high number, 50% of the people who get the flu vaccine actually find protection from it. I'm Jeremiah Kim, and I'm the co-founder of Signl, and we tune the immune system. So for vaccines, there are a lot of problems. For the patients, 77% of them feel negative side effects. People still get sick after vaccinations. You could think of this as fever, headaches, chills, soreness in your shoulder, and the other negative side effects that accumulate after vaccination. On the developer side, they also have problems, whether it's cellular response, durability, or the formulation of their vaccine. So what's the current problem in vaccine development? Our customers are vaccine developers, and they have a lack of control over the immune system. This creates vaccines that are lacking. Furthermore, this leads to markets that lack good products. So where does this lack come from? It comes from a lack of efficacy, a lack of durability, and these side effects that I've been mentioning. And this is not only true for vaccines, this is true for all areas of immunity. So how can we help? And apologies for the slides. It seems like the PowerPoint didn't transition correctly. This work is coming out of the lab of Professor Aaron Esther Khan, who is on the advisory board for the NIH, and myself, who is in his lab. We're joined by a robust team of scientific advisors and business advisors. I want to highlight Jeff Fairman in the top left, who is the founder of Vaxcyte, which is a $7 billion publicly traded vaccine company who's very interested in our work. From our lab, we have 5+ papers on the topics, 3 patents pending, over 18 years of vaccine research, and in total, $29 million in NIH funding to the lab, from which $2 million is available to Signl over the next 5 years. So what do we do in particular? There's a component in vaccines that creates adverse events, as well as creates the effectiveness of vaccines. What Signl does is add in a small molecule that is able to decouple control over immune response. This allows us to have a reduction in adverse events, but at the same time, increase in efficacy and durability and overall cellular response. One piece of data that I want to leave you guys with is this. In this particular experiment, we gave mice 1,000 times a lethal dose of flu. In every mice that had the normal flu vaccine, they died after 4 days, but every mice treated with our immunomodulators lived the full duration of this experiment. So what's our first shot on goal? Our current value proposition is that we could fix failed assets from vaccine developers by reducing side effects and increasing efficacy. So what this looks like is there are a lot of assets in pharma that wasn't able to pass forward across the finish line. These are significantly de-risked assets that couldn't pass phase 2 or phase 3 clinical trials, and we seek to push them across the finish line with the addition of our immunomodulators. So our resurrection pipeline will create new vaccine markets. These 3 different indications don't have any commercially available vaccines because of lack of efficacy or reactogenicity or negative side effects. So the inclusion of our compounds would be able to push it across the finish line. We're going to use the $25,000 from TechRise for de-risking experiments, particularly for our CMV model, as we want to push that as our first lead asset to the clinic. This will lead us to future milestones, such as an IND filing in 2027, which will cost around $5 million to get there. So we'll use the TechRise grant funds to help optimize these preclinical models and work towards making every vaccine better. Thank you.
Julia, can you start us? Julia, do you want to start?
Thank you for the presentation. I'd be curious to understand, in your clinical study, once you had measured the effectiveness of the immunomodulator on the mice, were you able to verify their resistance to the flu? Did that affect it at all?
Yes, so with that challenge model, we actually gave them the flu. So we had to culture the flu in the lab, and we administered it to them intranasally. So we've seen that they are able to create not just neutralizing antibodies, but protective ones. We also looked at the lung histopathology of those mice and saw that there was less inflammation in the lungs of those mice.
Wait, I'll add one more question. The immune system is extraordinarily complex. For the benefit of the group here, could you explain in simple words how your technology works?
Yeah, so in simpler words, you could think of vaccines or other stimulants to the immune system like a megaphone. So this just generates a signal that is loud and not really defined, What we're doing is adding in a sound mixer to this so that we could selectively tune up or tune down certain parts of the immune system so we could get a more customized response.
Thank you. So, Jeremiah, I think what's really exciting about this is the potential to help to bring vaccines that have failed before back to life, right? So, what is your strategy for accessing those vaccines?
Yep, so actually we are working really closely with the CBC here in Chicago to help identify lead indications. So we're in the process, we just got awarded the Director's Fund, and we're in the process of scaling up towards the Accelerator Award, but they've been helping us a lot with indication analysis and figuring out from different angles which ones make sense for a go-to-market strategy. Thank you, Margarita.
So related to that, the business process of securing rights to a failed candidate or venturing with, in many times, a startup company that has reached the end of its path because their vaccine got rejected earlier in the process. That's going to be a daunting business task. If you're all great scientists, but have you thought about how you're going to attack the business challenges of that?
Thank you, Robert. That's an excellent question, and we have thought about that a lot. So one of our main strategies for this is to identify ex-heads of these different pharma companies who kind of know where the bodies are buried so that they know that their product like 15 years ago didn't work because it wasn't that efficacious, or another product that they know of had a lot of negative side effects which resisted it from passing phase 1 trials. So we want to get in touch and have those people as our advisors and our leadership, our senior leadership, so that they will be able to guide those discussions and facilitate those agreements.
That was a little bit of my question, so I'm going to double down on it a bit. 90% of phase 2 vaccines have failed. You sort of know your field of opportunity if you're going after go-to-market in this way. Which ones are most interesting to you, and how do you make that decision versus just sort of going out and partnering with folks? What's the plan to identify, of those 90%, where you want to play?
Right, so that was what the top 3 vaccines that I selected in the slides 2 slides ago, CMV, C. diff, as well as Lyme disease. We've done a lot of analysis on different commercial vaccines that have failed recently, and we think that based off their mechanism of action, as well as why they failed, which is usually efficacy or these reactogenetic side effects, that our compound should be able to tune those systems in particular a little bit better than some of the other ones.
So as a follow-up then, are those from big pharma? Where would you then have to look to partner or get access to those specific assets?
They're from big pharma, yep.
Any final thoughts? You have a couple of seconds left. Any last questions? All right, Jeremiah, great job. Thank you so much. And last but not least, we have Anna Cornell with Acorn Genetics.
All right. Hello, everyone. I'm Anna Cornell, and today I'm excited to talk to you about the future of genetic sequencing. I started my journey 7 years ago, after my father was diagnosed with a serious genetic condition. I saw testing to see if I'd inherited it, but despite being connected to a plethora of sequencers from my lab and having great health insurance, there was simply no way for me to get a genetic test without spending $8,000 and waiting months for results. Frustrated, I started to talk to anyone and everyone who frequently needed access to sequencing and found that I was far from alone. Today's sequencers are large, expensive, complex machines that cost north of $1 million and require trained lab technicians in order to operate. As a result, all samples had to be sent to a centralized sequencing core in order to process. This frustrates researchers who are annoyed with the complex sample prep they need to do, makes carrier screening impossible for the 99% of specialty genetic disease clinics who can't afford the upfront $1 million cost of a sequencer, and ultimately leads to the destruction of 10 to 30% of agricultural yields every year due to delays in crop pathogen diagnostics. And so, a team of researchers and engineers from Northwestern University joined me in creating this solution, the Acorn Lab. While most sequencers are built to take 10,000 samples and look at 1 or 2 mutation sites, our sequencers are built to take 1 sample and look at 10,000 mutation sites. As a result, we were able to miniaturize the device, completely automate sample prep, and lower the cost of the sequencer to just $5,000. We're able to do this by using a novel nanopore solid-state sequencing chip, currently patent pending. Our sequencing chip uses a unique chip geometry, a bimodal signal processing algorithm, as well as the thermoelectric properties of a 2D material in order to accomplish long-read sequencing without compromising on speed or accuracy. So our competitors right now are all focused on creating better and better sequencers for wealthy research institutions who need to do high volumes of targeted sequencing, leaving the Acorn lab as the only viable option for labs, clinics, and research groups who need rapid, portable, and affordable sequencing. While our competitors are building the better and better supercomputers, we built the smartphone for sequencing. We further lowered the upfront cost of sequencers as well as the initial barrier to sequencing by adopting a razor and blade business model into the sequencing space, because we make the majority of our profit not off of the Acorn lab, but of cartridges used with every single test run. To put the numbers in perspective, we have about 100 pre-orders from clinics, labs, and groups across North America. By launching to just these 100 pre-orders, we will make about $250,000 from the sequencer, but $14.6 million in annual recurring revenue from our cartridge sales. But research clinics and labs are only the beginning. 7 years ago, I set out to get a genetic test for a condition that affected my family and found none. In the coming years, we plan to offer direct-to-consumer testing for the millions of Americans like me who need actionable, affordable, and accurate preventative health care. But our device isn't just a one-time use device. Because we have the ability to detect viral RNA and bacterial DNA, we have the potential to be the one-stop shop for detecting everyday illnesses in the home. The next time you're sick, take a cheek swab, throw it in the Acorn lab, and find out the exact strain of strep throat you have down to its antibiotic resistances without needing to leave your home. To date, we've raised our pre-seed and we've used to develop our Acorn lab, the alpha prototype of it. This upcoming fall, we're raising a seed round, which the $25K would hopefully come towards to help launch the Acorn lab to our initial 100 pre-orders. We are a group of Northwestern research and engineers, as well as a few directors we've managed to poach from other sequencer companies such as PacBio and Ion Torrent, with decades of experience in genetics, nanofabrication, and device commercialization, and are well poised to complete our mission of democratizing sequencing and putting an Acorn lab in every home. Thank you, and I'd love to take questions.
So sequencing is, the holy grail, right? Multi-million-dollar machines that you're reducing to,
$5,000.
$5,000.
Yes.
But for the consumer, understanding the value of sequencing could be a long process. Is there an intermediate market for adoption, like research labs or drug development companies that might validate the product and help make at-home testing?
Yep. So the people that we're launching to, I've thought extensively about this because my original goal was, let's go B2C. There's tons of people like me. And then for manufacturing reasons, other reasons, let's go B2B to people who are experts in the space. So our first customers are actually sickle cell disease clinics who are trying to run carrier screening in clinic but cannot because they can't afford the $1 million sequencer. So they have to outsource all that. They lose out on revenue. We're giving them a cheaper, affordable sequencer for them to be able to run these tests in-house. They are experts. They know the value of sequencing, and they can explain to their communities the value of a carrier screening test. Yeah.
Tell us a little bit more about the capital intensity of growing this business. How far will this raise get you, and how much capital would you need in order to get to scale?
Yes, perfect. So as of right now, we've been running incredibly lean, and we've been able to produce the first alpha prototype off of about $2 million total non-dilutive and dilutive funding. $25K would enable us to run our first pilots and would enable us to get the data we need to get the raise to launch the Acorn lab. Once we've launched, because we kind of have passive revenue through our recurring cartridge sales, we don't need that much further investor funding in order to get to a point in which we can pursue an acquisition or an IPO.
Okay, so I guess to follow up on that a little bit, so what is the regulatory path to this, and how long will it take for you to be able to actually
So no sequencers today are FDA regulated. They're all CLIA certified, which is a fairly straightforward regulatory process for us. It just basically means the device is correct and doesn't blow up. So we are pursuing that at the current moment. In terms of FDA approval, the only FDA approval you need in terms of sequencing is on the assays. So for us, we're currently supplying our assays through New England Biolabs, and they've already secured the FDA approval on the enzymes. So we're outsourcing them from them, putting them into our cartridges and running to the Acorn lab. We don't have to go through the FDA as a sequencer.
Okay, so how long would it take for you to then to get some sort of validation?
So we do have initial validation. Right? And we've run comparison studies at the Acorn lab with other, not the big three that I mentioned there, but we've run comparisons with Thermo Fisher and Allegiant, I think, or Agilent. I'm blanking on the name right now, sorry. We've run comparison, it's performed comparably, and we're seeking right now to publish our studies. Paper publications take much longer than initially anticipated, but we're currently in the process of publishing our research.
Nancy.
So I'll go into the premise that you've got some good head-to-head data, and you're looking for a faster, cheaper answer to put in the hands of others, and it's based on the nanopore gene sequencing.
Yes.
Can you give us 30 seconds in layman's of what that is and why it makes you so much more competitive of not only what's on the market, but what might be in the pipeline?
Yeah. So there are a couple other nanopore sequencer companies that are emerging. All of them are protein-based. Proteins degrade very quickly, and they're very expensive to replace. So our solid-state sequencer is long-lasting, so you don't have to deal with the constant $700, $800 weekly replacement rate. We're pricing our cartridges at about $50 per test. We make $40 off of every cartridge. And that's how we're making the majority of our revenue. In terms of how this carries on to the user, the cost of their test is whatever markup that the clinic decides to add on. So they're only going to be dealing with maybe a $75 to $100 test rather than having to go through an extensive long process and spend thousands.
All right, thank you so much, Anna. Great job. Great job to all of our founders. And now the judges have the judges have the very difficult task of deciding who's going to win $25,000. And while they're deliberating, you guys can leave the stage. Thank you. Go with Lucy. And while they're deliberating, we're going to have Tom join us and introduce Michelle. But Tom Denison is the co-founder and CEO and Chief Relationship Officer of SmartHealth Catalyzer. Tom leads the hub for Midwest biomedical venture creation, and SmartHealth builds and seeds funds. This copy is weird for me today. New ventures around most promising innovations discovered at Midwest universities and research hospitals. With over 25 years of experience in entrepreneurship, mentorship, and venture creation, Tom is passionate about building and funding biomedical startups that will transform and improve lives. And he'll be talking today with Michelle Hoffmann. Michelle Hoffmann, PhD, is the executive director of the Chicago Biomedical Consortium. That's CBC for the acronym people were talking about earlier, a consortium of biomedical researchers across Northwestern, the University of Illinois at Chicago, and the University of Chicago that is generously supported by the Searle funds at the Chicago Community Trust. The CBC's mission is to stimulate collaboration among scientists to accelerate discovery that will transform biomedical research and improve the health of humankind. In her time as executive director, she's recognized the CBC to train local PhD talent in the business of early science commercialization while developing a process to find and fund the most promising biopharma applications of Chicago's university science. And prior to her role, she was SVP of Deep Tech at P33 and spent 15 years helping life science companies grow, most recently as Senior Vice President at Boston-based Back Bay Life Science Advisors. Great, we're good. She did a lot of things. She's still very young. She also sold a company for $110 million.
If anyone wants to know my entire career, you can get me afterwards. I'll start from kindergarten. It'll be fascinating.
So I also know that Michelle is absolutely the hardest working, busiest person in the life sciences community here in Chicago. And if you want to meet with her, apparently you have to organize an event for 3,000 people to get her up here on stage. So anyway. By the way, also, I am not envious of the judges right now. Incredible 5 startups. Let's give it up one more time for each and every one of the founders. So, speaking of startups, put our startup caps on. I'm going to ask you a question that I know you and I want to ask initially of any startup, but let's talk about, and we're both very passionate about this, but let's talk about the problem that you and CBC are trying to solve.
Thank you for that. I'm actually trying to solve the problem that I never have time for lunch, and I have to do this to talk to you, Tom. That's my first problem. The second problem is that
We have a lot of university innovation here. What we don't have is capital, know-how, businesses. There are a few things that we don't have. And so we talk about translation, especially when we talk about life sciences. But the issue is that it's not even a valley of death. It's like a wide Grand Canyon chasm of like boiling fire that we need to get through in order to actually make companies that are investable. And that is what the CBC works on. I do want to point out that we don't just cover the 3 universities, we now cover 9 based on NIH funding as well. So pretty much any university, well, we fund 4 R1 and 5 R2 universities in Illinois, so if you have an idea, come on up to us. I want to say one other thing. You'll see some of the people on the stage refer to the CBC. We don't just give them money, and this is really, really important. We actually help them work through their scientific strategy, which is in fact their business strategy. And so we think that is what is needed here.
Got it. So I'm a big believer in this, and it's the premise of SmartHealth. Is that, we know there are amazing biomedical innovations coming out of incredible institutions. First question is, how do you go about identifying some of those amazing innovations? And then secondly, how do you go about identifying those aspiring bio-entrepreneurs that you are trying to nurture and inspire and educate?
So we do that 2 ways, or I'll answer your question, in terms of how do we find innovation and how do we select it? So when I came to the CBC in 2021, it had been around for a long time, and it was a very passive academic grant-making organization. Very, very standard, a bunch of professors, and this is I have to say I'm married to faculty, so anything disparaging I say about faculty is just from personal experience. But it was just a bunch of professors making guesses about what was translatable. And their idea of what was translatable is like what would save a mouse. And that is not, in fact, what will make something go into the clinic. So how do we find things? We basically revamped our process to run a lot more like a venture fund. And our basic gestalt is that academics need venture-like resources and expertise at the time when they cannot afford it. And even if venture is interested in them, they will extract, and again, no disrespect to my venture brethren here, but if it's too early, they will extract prices that are just not tenable. And so what we try to do is deploy our team of experts that are aided by our entrepreneurial fellows to do deep diligence on academic projects almost at inception. And as somebody that has 1 foot in academia, but who also spent 15 years in transaction advisory, helping build an organization in Boston, right? My observation has always been that the type of diligence that I would do on a live transaction is what academics need, but there is no way they could afford those prices. So really what we've done is take philanthropy and federal funding to build a SWAT team embedded in academia to work with faculty on a completely, we get no return, we take no equity. This is literally just to move things forward. That is what we do. And then the way that we do it is we have people like myself, we have my co-director Liz McMath, who also a scientist by training, spent time at Novartis, did search and evaluation for cell and gene therapies, and now Eric Schiffauer, scientist by training, drug hunter with Deerfield. And so we deploy our expertise, but we extend it by training PhDs who would otherwise have to leave Illinois for the coast to learn biotech, and we say, stay here, we'll pay you. You will be our extenders. We will train you experientially. You will get expertise like you will not find in any other fellowship. And then the goal is to train them for 2 years, and they commit to then going off into the ecosystem after their training. So this is how we do it, and this is how we train the next generation.
Wow, that's extraordinary. So can be a little bit delicate. How many academics are in the crowd here today? Any at all? Okay, good, one. All right, so just like your husband, well, not just like your husband, but
okay, just remember we're being videotaped. Okay,
okay, that's right. So my dad is also one of those. He's 84 years old, and he's in his 59th year of teaching and publishing and researching. So I've grown up around this, and again, I'm going to try to be a little bit delicate here, but we are talking about human beings, the scientists. So how do we go about sort of convincing, if you will, the scientists that your approach or my approach in sort of building companies around amazing innovations, what do we need to do to sort of engage more of the academic community to accept folks like you and I?
Okay, so again, some of my best friends are faculty. I was also raised by a professor. I was going to be a professor. All my friends are professors. This sounds like I'm about to just totally diss them. So I just want to say that I say this with context, and I also say in some ways that I was born for this job, right? I literally grew up in a faculty meeting. And what I will say is that I knew this going in, but my observations over the last 3 years is thus. University academic science is built on an individual. You have an individual lab who is getting funding, and everything is incentivized for that, right? So they need their papers, they need their funding, and they need it to be a single lab, a very specific expertise that they use to produce their product, which is not technology, it is papers, right? You need papers for everything. You need papers to get grants, you need papers to graduate students, you need papers for postdocs to get jobs. And so the fundamental thing that or whatever, my big insight, which is not that big of an insight, is that drug development is a team sport and academics is very individual. And so I don't believe necessarily that we need to be educating professors. I believe that if professors want to do this, then they should come to a resource like myself or you, and those resources should be, okay, now it is time for you to know that you don't know everything, and we are going to surround you with the expertise you need because drug development is hard.
Freaking love it.
And Desiree's about to cut us off, so.
Yeah, I know. So at that, let's give it up for
Sorry
Michelle.
to all the professors.
CBC, great to be here today with everyone. Thank you.
Thank you both for the really amazing conversation and helping educate us on all the incredible work that's happening across our universities and the collaboration that's happening. All right, with that, it looks like the judges are ready, so I'm going to invite the judges back on stage, and then all the founders, you want to come join us on stage too, so we can announce both the runner-up and the winner. So the runner-up matters because both the runner-up and the winner are in the running for the finale, which is a $100,000 non-dilutive prize that happens in November, which we hope you will join us for. And who's announcing today? Oh, great. Stephen Shaw, our partner at Verizon, is going to announce. Okay, so the runner-up.
All right, first to the founders, you all did a fantastic job. This was not an easy deliberation, especially in our time constraints, so you all should be proud of the work that you did. Given the tough deliberation, we still do have two winners. We have a winner and a runner-up. Our runner-up today from Signl is Jeremiah Kim. And that means, and Desiree explained the importance of the runner-up. And so the winner today of a $25,000 prize is Anna Cornell from Acorn Genetics.
Thank you so much. This is my first pitch competition that I've ever won after like 10, so this is, I'm so grateful. This money is gonna go right towards like getting these getting the Acorn labs into our initial pilot sites. I think we're going to be able to do a lot of carrier screening for sickle cell affected folks, and okay.
Well, I can't think of a better way to kick off TechChicago Day. So thank you all, founders, for your courage and for the incredible science that you're bringing to the world. And just so you all know, this is not a one-and-done experience for these founders. They can come back on another TechRise and win in the future, and that happens all the time. So we can't wait to support you on your journeys. Thank you so much to our sponsors today, Sterling Bay and team. Thank you to our incredible judges and to the incredible P33 founders team who makes all of this possible.
All right, everyone, we're going to take a quick break. We will be back here at 1:30 p.m. That is about 14 minutes from now. We'll be back at 1:30 p.m. on the main stage. You're going to love it. Right around the corner, we've got Dr. Mae Jameson speaking. She is an American engineer, physician, and the first woman of color to enter outer space. You're not going to want to miss that conversation. We're back here at 1:30 p.m. In the meantime, we have food trucks out in the driveway in front of the building. There's a lot of action going on the floor beneath us, on the 7th floor, the AnitaB.org celebration stage, the Grace Hopper celebration stage. There's content there. There's some breakouts happening on the 7th floor. There's also a co-working lounge if you need to get some work done today, happening on the 1st floor, courtesy of Workbox. We'll be right back at 1:30 p.m., everybody. Thank you.